Good evening everyone, I'm Coach Spivey and we're back once again with another amazing science tutorial video. And in this video we're going to be going over mitosis. But before we get started, I want to give a shout out to the following students. So let's start off with Makia Gainey, we have Jakiria Norman, Dylan Quick, Antavia Roberson, Denzel Jones, Danae Hammond, LaDeja Dawkins, Paradise McGlown, Nakiria Albritton, and Nadia Lucas. The following students I just called said, Coach Spivey, we need you to make a video on mitosis and break it down so we can understand. So ladies and gentlemen, I hope this video is helpful. I appreciate all the feedback that you gave me. So let's go ahead and get started. So let's take a look at mitosis. And mitosis is asexual reproduction, which means that an organism is actually making copies of its cells. Now, asexual reproduction occurs in prokaryotes because they don't need a partner in order to reproduce. So they literally make copies of their cells. But in organisms like humans, we go through mitosis in order to make copies or to make new cells for our bodies. So mitosis is the process in which the nucleus divides to form two new nuclei. And mitosis begins after a sperm fertilizes an egg in humans and in other organisms. So we look at the phases of mitosis. We start off with interphase, then to prophase, then we move to metaphase, then to anaphase, and then to telophase. It's genetically identical daughter cells with the same amount of DNA as the parent cell. So first, there are 46 chromosomes in the parent cell. And then the DNA is replicated during the S phase of the cell cycle, which gives us 46 chromosomes and 92 chromatids. The end result of mitosis is two genetically identical daughter cells with the same amount of DNA as the parent cell. So if you notice, the two daughter cells contain 46 chromosomes, which is the same amount of chromosomes as the parent cell. There are three reasons why mitosis or asexual reproduction occurs. One, for growth, so for our bodies to develop and grow over time. Two, for repair, so when we get cuts or scrapes or bruises, or when we break a bone, our bodies need to replace those cells so our cells can heal. And then three, for replacement. Our cells get old over time and they need to be replaced with fresh new cells. So we take a look at this. As we're younger, our body replaces and repairs those cells, and they continue to happen at a rapid rate when we're younger. But then as we get older, the process of mitosis begins to slow down. And so instead of those new cells getting replaced rapidly and quickly, they start to get replaced at a much slower rate. If we take a look at the cell cycle, if you notice, most of the cell cycle is in interphase. So in interphase, we have G1 phase, which is going to be cell growth. And then we have the S phase. This is where our DNA gets replicated or a copy of our DNA is made. And then if you look at the G2 phase, the G2 phase actually prepares for mitosis. The part that we're going to focus on in the cell cycle is going to be this mitosis part right here, where the cell actually makes a copy of itself. And then if you look in this M phase with mitosis, we have prophase, metaphase, anaphase, telophase, and then the last part is cytokinesis, and then the process starts all over again. The phases of mitosis are in the following order: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. And I have them listed in order on the side over here as well. And a way that you can remember it is, I came up with my own mnemonic and I said, I protect my Aunt Tina's children. So this is an easy way you can remember the phases of mitosis. So I protect my Aunt Tina's children, which stands for interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Now for your first check for understanding. And you're going to put the following phases of mitosis in order. You have 1 minute to put them in order, and I'll pause the video starting now. So we take a look at it. Interphase is our first phase of mitosis. And in this phase, we have cell growth and DNA replication. So the cell is actually growing, getting larger, and then DNA is being replicated, or a copy of the DNA is being made. So if you look at an animal cell, here's the interphase for an animal cell, and then in a plant cell, here's the interphase for a plant cell. Remember, the cell is growing and DNA is being replicated, or a copy of it is being made. Now let's move to prophase. And in prophase, the chromosomes appear condensed. So here are our chromosomes right here. And when we say condensed, that means they're getting close together or getting tightly packed together. And it says, and the nuclear envelope is not apparent. So here's our nuclear envelope right here. And if you notice, there are gaps in the nuclear envelope. Why are there gaps? Because this nuclear envelope is dissolving or disappearing. So if we take another look at prophase, chromosomes condense and the nuclear envelope disappears. So in an animal cell, notice the chromosomes are condensing or getting tightly packed together, and then the nuclear envelope is disappearing. Same thing in a plant cells, the chromosomes are condensing and the nuclear envelope is disappearing. Now let's take a look at metaphase. In metaphase, the thick coiled chromosomes are lined up in the center of the cell on the metaphase plate. So here are these chromosomes that are lining up in the middle of the cell. The spindle fibers are attached to the chromosomes. So here are our chromosomes lined up in the middle, and here are our spindle fibers attaching to these chromosomes. And these spindle fibers are attached to the centrioles at each end. Here's another look at metaphase. So the chromosomes line up in the middle and the spindle fibers attached to the chromosomes. So if you look at an animal cell, here are our chromosomes right here, and those spindle fibers are attaching to them. Then if we look in a plant cell, here are our chromosomes lined up in the middle as well. And it's very hard to see, but our spindle fibers are attached right here as well with the plant cell. Now let's take a look at anaphase. In anaphase, the chromosomes have separated and are moving toward the poles. So if you look closer, those chromosomes that were lined up in the middle during metaphase have now been pulled apart with these attached spindle fibers. These spindle fibers are pulling apart these chromosomes towards the centrioles located on both ends of the cell. Here's a closer look at it. So in anaphase, the chromosomes separate and move to the end of the cell. So if you notice in this animal cell, here are our chromosomes, here are those spindle fibers attached, and here are those centromeres pulling them to each end. Same thing on this side as well. Here it is in a plant cell. Here are our chromosomes on each side. We have those spindle fibers attached, and then we have our centromeres pulling them apart on each end. Next we move to telophase, and in telophase the chromosomes are at the poles and are becoming more diffuse. And what do we mean by diffuse? We mean that they are actually separating apart. If you notice in metaphase, prophase, metaphase, anaphase, and telophase, they're becoming condensed and becoming tightly packed. Well, if you look in telophase, they're actually separating apart from each other. And then it says the nuclear envelope is reforming. So if you take a look, here's that nuclear envelope forming back around our chromosomes. And then it says the cytoplasm may be dividing. If you look in the middle, this is where the cell starts to divide. So here's another look at telophase, and it says the chromosomes are at the poles, the nuclear envelope reforms, and the cell begins to divide. So you can tell right here, here's that cell division right here. Our chromosomes that were tightly packed, they started to diffuse or they start to spread apart from each other. So the same thing in this plant cell. Here's that cell division that begins to occur or start to happen. And if you notice our chromosomes, they start to become less tightly packed or start to separate. And then our last step is cytokinesis. So the cell splits completely and two new cells are formed. So if you notice, the cell has completely split. They're no longer attached to each other. The nuclear envelope is reformed around the chromosomes, and we have two new cells. Now let's take a look at a mitosis animation. So if you notice that nuclear envelope disappears in prophase, and in metaphase they line up in the middle, anaphase they pull apart, and then in telophase you have the nuclear envelope reforms and its cell begins to split. So let's look at it again. We have prophase, chromosomes condense, nuclear envelope disappears. Metaphase, the chromosomes line up in the middle. Anaphase, the spindle fibers pull it apart, pull the chromosomes apart, and then we have telophase right here. Cell splits and divides in cytokinesis. One more time. Prophase, chromosomes condense, nuclear envelope disappears. Metaphase, chromosomes line up in the middle, attached to these spindle fibers. Spindle fiber pull it apart in anaphase, and then in telophase, that nuclear envelope reappears. Then cytokinesis, that cell splits apart. So now, ladies and gentlemen, you're going to do another self-check or another check for understanding. You have 1 minute to label each part of mitosis in the following pictures, and you can pause the video and get started now. So now let's take one more look at the cell cycle. First, we start off with interphase right here, and this is where cell growth and DNA replication occurs. And then we move on to prophase. In prophase, the DNA condenses into chromosomes, the nuclear envelope disappears, and spindle fibers form. Then we move to metaphase. Those chromosomes line up in the middle of the cell, and they are attached to these spindle fibers on each end, which are attached to these centrioles at the ends of the cell. Then we move to anaphase, and in anaphase, these chromosomes are pulled apart and separated to opposite ends of the cell by these spindle fibers, which are connected to the centrioles at the end of each cell. Then in telophase, which is the final stage of mitosis, the cell begins to separate and divide. The nuclear envelope begins to reform around the chromosomes. And finally, in cytokinesis, the cell is completely separated into two daughter cells with the same amount of genetic material from the parent cell. The nuclear envelope is fully formed and the chromosomes have been uncondensed back into DNA. So now for your last check for understanding. In this check, you're going to write each phase of mitosis based upon its order and picture description. Ladies and gentlemen, I'm Spivey, signing off with my son Jordan Spivey, and make sure you have an awesome, wonderful, positive day. Peace.